Okay, good evening everyone. We are going to start the third session for the PMRF-NPTEL classes on Introduction to Dev Biology. Okay, let's do this. Okay, so as you would know, these are the PMRF-NPTEL sessions on the NPTEL course Introduction to Developmental Biology. Today is the third week and we are going to go into some basics of molecular biology, of how transcription and translation regulation work, as well as a bit of genetics as well. So these are problem solving sessions in which we go through the quizzes of the past year's run of the course so that we can have a better idea about to solve quizzes of this year's course, as well as have a better idea about the concepts behind the questions and the course material. I will briefly introduce myself. My name is Anuvab Chatterjee. I am a PMRF fellow at IIT Kanpur and I will be taking this course. So, how we do the course normally is that I show a question and then I ask people to actually try and solve it and give me an answer, and then we go through the concepts behind the question and then show the answers and we check what uh, what the correct answer is. Okay, so on to the first experiment or the first question. The question is, for a particular mRNA, a northern hybridization experiment. So northern hybridization experiments, if you would know, are experiments in which you probe for different RNA or mRNAs transcripts present in a particular cell. So this is similar to a southern blot or a western blot, but just for mRNA. And a northern hybridization experiment was carried out using the total RNA isolated from cleavage state and gastrulation stage embryos. Hybridization to intron specific probes indicated identical levels of mRNA in both stages. However, an exon specific probe indicated levels in gastrula to be fourfold more than in the cleavage stage. This is due to, and the options are, differential nuclear export of the mRNA, differential stability of the mRNA in the cytoplasm, selective inhibition of mRNA translation, and differential splicing of the mRNA. So to just clarify here, when we say it is an intraspecific probe, that is obviously going to be in the case of nuclear RNA, which has not been yet completely processed to a mature mRNA, and exon specific probe actually can span the exon junctions. So whenever the splicing has occurred, only the exon specific probe will detect those mRNAs. So mRNAs which are mature, which have the splicing done and are probably present in the cytoplasm and are about to go into translation, those are the ones which are going to be caught by the exon specific probe, whereas the intron specific probe will catch immature mRNAs which are present in the nucleus, which have not undergone splicing yet. So it seems that the particular mRNA we are looking at has fourfold more levels in the gastrula stage rather than the cleavage stage, even though their initial immature nuclear mRNAs were probably the same level. So what is the reason behind it? And the options, as I said, are these four: differential nuclear export of the mRNA, differential stability of the mRNA in the cytoplasm, selective inhibition of the mRNA translation, and differential splicing. Okay, I will give you a second to try and guess what the answer is. Okay, then we look at this, and this is the example on based on which the question has been asked. So this is a radio labeled uh, RNA in situ hybridization, in which we can see the two different tissues here, and these labels show wherever the mRNA for the Cy3A gene is present. And then here is the northern blot, in which if you can see that the total RNA extracted from the ectoderm and all the endoderm and the mesoderm are run separately. And as in the question, we see that in the case of an intraspecific probe, both levels seem to be similar in ectoderm and endoderm or mesoderm. And in case of the exon specific probe, however, the ectodermal Cy3A is much more uh, compared to the endoderm or the mesoderm. So this is exactly what the question is, and now we want to know why. So this can be for multiple reasons. The first reason could be that there is selective transport of the mRNA from inside the nucleus to the cytoplasm, and that can cause this. Or it could also be differentials. It could also be differential stability of the mRNA in the cytoplasm, in which case you just degrade the mRNA whenever it is coming out of the nucleus, and you just immediately degrade it so that it does not go and start translating proteins. So that is why the level is low. So it could be differential nuclear export, so none of the mRNA is being spliced and exported, or differential stability of mRNA. So in the case of the ectoderm, the stability is much higher, so much more mRNA is accumulating there, whereas in case of the endoderm or the mesoderm, it is immediately being cleaved off, and so you do not have a large amount. Okay, so the correct answers were A and B, and we are moving on to the next question. The question is, which of the following may result from mutations in cis regulatory elements? The options are change in localization of mRNAs, inability to bind splicing enhancers, inability to bind RNA polymerase, misregulation in splicing, synthesis of truncated proteins, inability to bind transcriptional factors. So we have a lot of options. You have to choose which of the following may result by mutations in cis regulatory elements. Okay, let us look at the answer. So first we have to know what the difference between the cis and a trans regulatory element is. A cis regulatory element is present on the same chromosome and is tightly linked to the gene in question. So all the time what cis regulatory elements are, these are DNA sequences that are present either upstream or downstream or within the gene, which affect the transcription or any like the expression of the entire gene. So DNA elements present on the same chromosome linked closely with the gene and has effects on the gene, whereas trans regulatory elements are diffusible factors which are not present on the same chromosome, are coming from somewhere else, and not linked directly to the gene, but they diffuse to the gene and do their function and regulate the gene. So here is an example where say on the red line, this is one chromosome, this is our gene, the blue, and this first yellow dot is the cis regulatory element which directly affects the regulation of expression, whereas the trans element which is present on a different chromosome or very far away, this actually forms a product which diffuses and this then affects the gene expression. So examples of cis regulatory elements are very abundant. It could be enhancers, it could be transcription factor binding sites, it could be silencers, and it could be present all over the DNA sequence close to the gene, tightly linked with it, whereas everything else which comes from somewhere else is a trans factor. So examples include the actual transcription factors. These are proteins which are not present near our gene, could be somewhere completely different and come in and help in regulation. It could also be different microRNAs which inhibit uh, for the translation or help in degradation of the mRNA and thus help in regulating the expression of the gene. So anything which is coming from somewhere else is trans acting. Anything which is present on the same segment of the DNA can directly help or regulate the gene expression is a cis factor. So the question was, which of the following may result in mutation is cis regulatory elements. So as you imagine, cis regulatory elements are essentially the entire DNA sequence. So any kind of mutations can cause a lot of changes. So changes in localization of mRNA. So localization of mRNAs is something that we will look at in a different slide. Is when mRNAs are transported to different parts of the cells where they are kept ready to be translated, and these mRNAs contain a localization signal on them, and without, if these cis regulatory elements are mutated, then the mRNAs cannot go where they are needed, and that will cause a lot of problems. Our splice inability to bind splicing enhancers. So there are particular splicing enhancer binding sites present which are recognized by splicing enhancers to actually splice the introns from the immature or nuclear mRNA, and if these are mutated, then you will have different splicing variants which will not probably be functional. Inability to bind RNA polymerase. So if the transcription factor B binding site, which is the B sequence, is mutated, then TF2B will not bind and it will not bring RNA polymerase with it to bind. And this also is changed in the DNA sequence. So cis regulatory element mutation. Misregulation is splicing. The same thing, if splice donor or acceptor site is mutated, then you do not have the correct splicing, and then you have misregulation in that. Synthesis of truncated proteins. So in the gene, if you have a mutation in which you have a stop codon being present where it should not be due to a mutation, then you will have premature truncation of the protein because the ribosome will fall off, it will not make the entire protein, and you have a truncated protein. And finally, inability to find bind transcription factors. So wherever there are enhancers, if you have mutations in them and transcription factors cannot bind, then you will have inability to bind to transcription factors, and then problems in regulation of the gene. So essentially, cis regulatory elements do a lot of things, and all of these are just part and parcel of it. So any mutations will cause a lot of problems because it can mess up regulation in various different ways, as we just saw. Okay, we move to the next question. Which of the following statements regarding gene expression are true? The questions are, the options are: a paternal imprint allows expression from a paternal allele. Alternate 5' and 3' splicing are mutually exclusive. Alternative splicing is the main mechanism by which protein isoforms are generated. Protein isoforms may have opposite effects, and a mutation in the 5' splice site prevents binding of U1SN RNA. Okay, I will again give you a minute to think about this. Okay, we are back. Let us look at it. First, we are going to discuss part of alternate splicing. So alternate splicing is actually the main source of variability in different proteins that are functional isoforms of proteins that are made. So as you would know that C. elegans, which is a nematode and which is like really small, has the same amount of genes that we have, or like similar amount of genes that we have, but we are hugely more complex both anatomically and functionally, because we have a lot of alternate splicing to make different functional variants of different proteins, which then add to the complexity of the whole system. So let us look at how alternate splicing changes things. So alternate splicing is when you have changes in splice variants. So during splicing, with the not only the introns can be specifically removed, but which exons are included in the final protein, or which parts of the exons are included in the final protein, can also be is also part of alternate splicing. So the first example we will look at is when we have alternate 5' splice site. So when we have an alternate 5' splice site, as you can see here, this two dashes here. So what this shows is that the exon 1 will go, the splicing will occur from this intron to this intron. So 1 to 2 is fine, but in case of 2 to 3, two different 5' splice sites can be used. One which is at the end of the exon, just at the start of the intron, which is the normal one, where this to this V section of the intron is removed, and so you will have one protein BCLXL, or one splicing variant in which the entirety of the second exon is included, as you can see right here. And this uh, yeah, and then the other case, in the other case, there is a 5' splicing site also present in the middle of the second exon. So it could be that this entire huge portion of DNA is just spliced out. So it not only splices the intron but also part of the exon, and so you have a shorter protein. And this is really important because one of these guys are pro-apoptotic, or makes sure that the cell undergoes apoptosis or programs cell death, and the other one actually inhibits apoptosis. So exactly, just alternate splicing can cause two different variants of the protein which have completely different functions present, arise from the same genomic sequence. And as we are talking about the functional diversity, or the functional different functional variants that the protein can form, this is the best example for it, which is known as discam. Discam is a protein present in discovered in Drosophila, and here uh, in case of discam, what happens is that it has a huge variety in which exon will be finally included in the mRNA, and then finally in the protein. What I mean by that is that, as you can see right here, there are 12 alternative exon 4s that the discam uh, gene can choose to actually include in the mRNA. So it will go all the way from the exon third exon, and it will choose one out of 12 of the exon 4. And then similarly exon 6 has 48 different alternatives. Only one will be included, it will be chosen depending on the situation. As well as exon 9 has 33 different alternatives, and exon 17 has 2 alternatives. So depending on which one of these exon cassettes are chosen, you will have different variants. And it has been shown, like if you just look at the numbers, then there are 38,000 different uh, variants of discam that can be produced. And that just shows how incredibly different variants can be produced from just one genomic sequence. Just choosing how the alternate splicing occurs can give us so many variations. So yeah, which of the following statements regarding gene expression is true? The options are: alternate splicing is the main mechanism by which protein isoforms are generated, which we saw. Protein isoforms may have opposite effects, which we also saw in case of BCL proteins. And a mutation in a 5' splice site prevents binding of U1 snRNA. So that you would know that the U1 snRNA is one of the parts of the entire spliceosome complex, which binds to the 5' site and helps in detection of the 5' site. So if there is any problems with the 5' splice site, such as mutations which prevent U1 snRNA to recognize the particular thing, U1 snRNA to recognize the particular sequence, then it will there will be no splicing on that site, and thus problems occur. So that is also the correct option. Okay, moving on. The next question is select the correct statements regarding the regulation of mRNA. The options are 5' methylation of unmethylated mRNAs aids in achieving translational competency. Poly-A tail length affects mRNA stability. 3' UTRs are not required for mRNA stability but are required for translational regulation. Cis-acting proteins are bind to splice sites and promote splicing. And masking loops the mRNA by binding to proteins bound to 5' and 3' ends of the mRNA and prevents translation. So which of the following do you think is the correct option regarding regulation of mRNA? You can just look and choose. I'll give you a second. Okay, let us look at it. So this we are discussing about how translation regulation occurs. So normally when we draw our mRNAs, we always draw them in a straight line with different elements present. But in the cytoplasm, they do not exist as a straight line, but instead in this circularized form with a crown on top of it. What is this? So in case of cytoplasmic mRNAs, they are bound by multiple proteins. When these cytoplasmic mRNAs are dormant, they are bound by two different proteins. One is a 4E protein which binds to the 5' cap, and then there is another protein which is known as CPEB which binds to the 3' UTR, and both of these are bound by the protein maskin. Whenever maskin is bound to both of these proteins, then translation is inhibited and you have translationally dormant mRNAs. But when this mRNA has to become active and start translating, then a different protein known as PABP, which is poly-A tail binding protein, binds to the poly-A tail. No, first what happens is that there is this protein called CPSF which comes in, phosphorylates CPEB, which moves masculine out of 4E. So you see masculine was bound to 4E and now it is removed completely. Once it is phosphorylated, CPSF binds and recruits the poly-A polymerase. So some mRNAs have a poly-A tail added to them before being exported out of the nucleus. Some after they come into the cytoplasm, poly-A tail is added. So in this case, the poly-A tail is being added in the cytoplasm, and once the poly-A polymerase starts adding the A tail, this is recognized by the protein PABP, poly-A binding protein. And once poly-A binding protein binds to the poly-A tail, it recruits 4G, which interacts with 4E, and which in turn gets the ribosome to bind and then start translating down the mRNA. So maskin bound mRNA prevents any translation. Whenever the BP binds, then it removes maskin and then you have translation. Okay, and this we are discussing because one of the other options was that poly-A tail is not required for stability. So this just shows how miRNAs work, and this we are going to discuss in one of the upcoming slides as well. So miRNAs or microRNAs are small RNAs produced by the different parts of the genome. These normally do not code for proteins, they only code for the small RNAs. What do these RNAs do? The microRNAs, after being processed, can be used to recognize different parts of a particular mRNA. So they go and bind majorly to the 3' UTR, and when they bind to it, they can prevent translation. So they are a translational inhibitor. So when a microRNA binds along with, so it is called miRNP. So it is microRNA ribonucleotide protein complex, because it is just not the microRNA but a lot of other proteins are there as well. which bind to the 3' UTR, and they can initiate a block to the ribosome and prevent the initiation complex from even getting the ribosome there. It can start degrading the mRNA poly A tail, which reduces the stability of the mRNA and it is degraded, or it can start directly degrading the nascent peptide by adding degradation signals to the nascent peptide, so that whenever it is translated and goes out, it is immediately degraded. So it is a huge part of the translational inhibition and regulation part of a gene expression. So it binds majorly to the 3' UTR, prevents translation by these three methods. Okay, so coming back to our question, select the correct statements regarding regulation of mRNA. Okay, so the options are 5' unmethylated mRNA, 5' methylation of unmethylated mRNAs aid in achieving translational competency. So this is just because in case of eukaryotes, the ribosome binding site is the 5' cap. In case of prokaryotes, it is a sequence known as Shine-Dalgarno sequence, but in case of eukaryotes, it is just the 5' cap. So if the 5' cap is not there, then there will be no translation because the ribosome won't sit on it. So 5' cap is absolutely necessary for translational competency. And this is why eukaryotic mRNAs, or eukaryotic genes rather, are not multicystronic. So in case of so you have one gene and one mRNA, and one mRNA forms one protein, but because there is only one site where the ribosome can bind, and it goes to the start codon and then falls off at the stop codon, so it can only translate one open reading frame. But in case of prokaryotes, since the ribosome binding site is known as is a DNA sequence, the Shine-Dalgarno sequence, then if you have multiple Shine-Dalgarno sequence, then you can actually have multiple different open reading frames on the same mRNA. So prokaryotic genes can be multicystronic. So you make a huge mRNA which has different open reading frames. One ribosome binds to one Shine-Dalgarno sequence, goes from start to stop codons, and then falls off at the stop codon. A next ribosome binds to a different Shine-Dalgarno sequence and repeats the same process. So the same mRNA can form multiple proteins. So multicystronic in case of prokaryotes. In eukaryotes, there is only one side where it can bind. There can't be more than one 5' cap, because that is just how it, like there is only one 5' end to the entire mRNA. So it is monocystronic. Okay, coming back to this, the next option is poly A tail length affects mRNA stability. It obviously does. There are different nucleases which are known as exonucleases, which go around the entire cytoplasm. These guys start chopping up RNAs from one end. So the 5' end is protected by the 5' cap, because that bond cannot be broken by these nucleases, but they can bind to the 3' end and start chopping. And that is why the poly A tail is important, because a large poly A tail means whatever exonuclease comes and tries to chop the RNA off will be thwarted by the poly A tail, because it will chop just a small portion of the poly A tail, and the entire information of the mRNA will still be safe and can still go on translating. And the last option, which is correct, is Maskin loops the mRNA by binding to the protein to the 5' and 3' ends of the mRNA and prevents translation. This is just what we saw. Maskin binds to both 4G as well as CPEB and forms a circular RNA which is translationally dormant. Okay, so that was this question. So we are moving on to the next one. So the question is, identify the correct statements regarding small RNA mediated regulation of gene expression. The options are: complete complementarity of the microRNA with the target sequence enables selectivity. MicroRNA's primary target 3' UTR of an mRNA. RNAi is required for endogenous genes but not for exogenous genes. Argonaute protein is responsible for binding to the target mRNA and inducing cleavage. And final is the seed sequence complementarity to the target resides in the 3' end of the microRNA. So so I will let you guys look at this, and then if you want you can try and answer this and put whatever you think is the correct options into the chat. I'll give you a minute to try and solve this. Okay, so since we do not have an answer, we'll look at it. So this is a brief description of how microRNAs work. So as we have been discussing, microRNAs are present on the genome and do not code for actual proteins but rather code for small RNA molecules. So once a microRNA gene is transcribed, it forms something called the pre-microRNA, which is this long hairpin loop structure with ends to it. These ends are chopped off by the nucleus drosha, and you only have a hairpin now, which is known as the pre-microRNA. This pre-microRNA hairpin is then exported and then incorporated into the protein complex known as dicer. Dicer contains multiple proteins in eukaryotes, one of them being argonaute protein, which binds to the uh, no, not dicer, but dicer, what dicer does is that it chops off the hairpin loop, and so you have two parts of it. So one of the parts is then incorporated into the risk complex. This risk complex has multiple proteins, one of which being argonaute, which actually binds to the microRNA that is formed right here, or the part of the microRNA here. And now the risk complex will go around scanning different mRNAs. Whenever the RNA sequence here is complementary to mRNA present anywhere, then the risk complex will raise a flag and it will stop the translation. So the risk complex sits on this mRNA, and in case that there are mismatches, so you see there are loops, which means there are mismatches here, a, or like here, then it means that the microRNA that was transcribed is not one-to-one match or completely similar to the sequence, and in that case only repression of translation occurs, in which the risk complex prevents the ribosome from actually going and transcribing the mRNA. But in some cases there is complete complementarity, so there are no loops to show that, and if mRNA binds something like this to the risk complex, then it causes a cleavage of the mRNA. So, but in both cases, if a microRNA binds, as we saw last time, it will either cause repression of translation or direct cleavage of the mRNA, preventing any further translation. Okay, so which are the correct options? The first one is microRNAs primarily target the 3' UTR of mRNA. So the 3' UTR is the portion of the mRNA which is after the stop codon, which will never be translated because the ribosome falls off at the stop codon. But this 3' UTRs are really important for regulation, because 3' UTR regions are very specific to a particular mRNA. So microRNAs actually only detect this 3' UTR to make sure one mRNA is repressed or not. This comes in handy in complex eukaryotic systems where we have multiple copies of very similar genes or family of genes, such as Hox genes, Hox A. There are multiple, so there are multiple Hox A genes themselves, so Hox A 1 to 13, and then you have Hox B, Hox C, Hox D, a lot of Hox genes. So many of them have sequences which are similar across them because they are the same family of proteins. So if the cell wants, say, I want to stop HOX A3 from being translated, but I need HOX A5, how will you regulate it with such a precise manner, because the sequence is very similar? So what you do is that you do not go after the protein coding sequence, the open reading frame, the ORF, because those will be similar because these are same family of proteins. Instead, you go for the 3' UTR. So all the 3' UTR are very specific to a particular variant. So HOX A3 and HOX A5, even though the protein sequence is really similar, the open reading frame sequence is very similar, the 3' UTR region will not be similar. And based on that, you can regulate it, and thus most microRNAs bind to the 3' UTR region of the particular gene and a particular mRNA, and not the open reading frame. But there can be cases in which they even bind to the open reading frame at the 5' UTR, but most of the cases 3' UTR. So microRNAs primarily target 3' UTR of an mRNA. And there are also situations in which you can, so based on the microRNAs and later on with multiple discoveries during the early 2000s or the start of the 2000s, there was this one scientist known as Andrew Fire who discovered something called RNA interference, which essentially describes how microRNAs work, and later on people adapted their work in order to make the RNA interference system, in which you make artificial microRNAs which you put into cells and make sure that some particular gene is not active. So if you want a particular gene, say HOXA2, to not be active, you make a microRNA against that and cause the cell to take that particular microRNA up, and once it has taken up, it will go through the same process that we saw, go through DROSHA, then dicer, then RISC complex, and then it will sit down on the protein of your choice or the mRNA of your choice and will start repressing its translation. And since you can choose the sequence by which you will make the microRNA, you can also choose how which proteins you block. So this is regularly done in molecular biology, like we also do it a lot in our lab. And the other one is the Argonaute protein is responsible for binding to the target mRNA and inducing cleavage. And this is also true because, as I said, the part of the RISC complex, so RISC is a huge complex which has different proteins. Argonaute is the one that is responsible for binding to the microRNA, binding and complement. So it determines the complementarity between the microRNA segment and the mRNA, and it is the one that, since it is binding to the microRNA, and then it goes along the mRNA and sees if it is complementary, and then if it is, then the entire RISC complex then cleaves the entire mRNA and prevents any translation. Okay, so that was this question. We move on. The next one is identify the correct statements regarding the control of gene expression. The options are: transcription factors are dispensable in prokaryotes, enhancers increase the rate of transcription binding to regulatory sequences, HDACs or histone deacetylases recruited by transcription factors result in reduced transcriptional activity. Transcription factors are transregulatory factors but not microRNA. The transcription elongation complex releases RNA polymerase II from the mediator complex. An enhancer of a gene can bind only to one transcription factor, while a transcription factor can bind to multiple enhancers. Okay, I will put this here. So you have to choose which of the following you think is the correct option for gene expression. I will give you a minute to think about it and put the correct option into the chat. You can put A, B, C, D, E, F and choose whichever you think is the correct option. Okay, so let us look at it. So here the first option, transcription factors are dispensable in prokaryotes, is true. So in case of prokaryotes, it works out a bit differently, or rather completely oppositely to how it works in eukaryotes. In eukaryotes, most of the genes are in the off position, so there is no transcription in most genes to begin with. Only when enhancers are bound by transcription factors do we have someone answering. So they have said 1, 4 and 5. We'll go through it in a bit, which ones these are. So transcription factors in eukaryotes, so in eukaryotes, whenever the transcription factors binds to an enhancer, then only transcription is initiated. But in prokaryotes, the situation is completely opposite. In that case, in case of prokaryotes, the genes are always in the on position. All the genes are being transcribed all the time. So transcription factors actually do not play much of a role other than inhibiting the transcription. But enhancing doesn't work that way because all of it is being transcribed most of the time. The next option which is correct is histone deacetylases recruited by transcription factors result in reduced transcriptional activity. This part we have actually gone through in the last week of the lectures, in which we learned that histone deacetylases actually remove the acetyl group from the histone molecules, which bind the DNA. So once the acetyl group are removed, it causes the histones to compress and prevent, so they convert to heterochromatin, in which transcription factors cannot bind, and so you have reduced transcriptional activity. And final correct option is the transcriptional elongation complex releases RNA pol 2 from the mediator complex. This is also true. So, basic molecular biology. The mediator complex is the one that causes the RNA polymerase 2 to come and bind to the promoter site, but after that the transcription elongation complex takes over and causes the RNA polymerase 2 to move around, move down the DNA segment to actually start transcribing. Okay, so the particular message that we got was that options 1, 4 and 5 are correct. So 1 is correct, that is true. 4, transcription factors are transregulatory elements, and but microRNAs are not. So when we are going through a previous question, I explained what a cis and a transregulatory element are. Cis regulatory elements are DNA sequences present really close to the gene of interest and are tightly linked to it. So something like a transcription factor binding site, or a localization signal, or an enhancer, or the promoter itself, cis regulatory elements. Transregulatory elements are the ones that are not linked to the gene of interest, so it can be very far away on the same chromosome, from a different chromosome, exogenous, whatever. And so transcription factors fall into that category because they do not need to be near or cis. They are from somewhere else. They can be transcribed by a gene from a different chromosome. It makes the protein, and the transcription factor comes and binds to the transregulatory sequences. So yes, it is a trans factor. But microRNAs also fall in the same category. They need not be close to the gene at all, could be in a different chromosome, and being transcribed there, it comes and finds the mRNA of the gene and then causes its degradation or inhibition of translation. So microRNAs diffuse from a different place as well, and are not necessarily around the gene of interest, so they are also transacting factors. So anything coming from out somewhere other than the DNA sequence close to the gene of interest is transacting. So this is not correct, and the 5 is, yeah, 5 is the correct option. Okay, moving on. Identify the correct statement regarding translation and post-translational regulation. The options are: mutations in the Kozak sequence will hinder efficient translation. Deletion within the PRIB-now sequence will affect histone modification. Poly A binding protein PABP binds to poly A tail and promoter translation quiescence. microRNAs cannot affect the translation of mRNAs bound to the ribosome. mRNAs localized to specific regions within the embryo can be selectively translated. Okay, so I will give you a minute to try and answer this and try which is the correct option. Okay, so coming back, let us look at it. Okay, the first we look at the Kozak sequence. The Kozak sequence is actually a consensus sequence that is present in the mRNA, and it is actually the sequence which determines where the translation will start. So just the start codon, by just the start codon, as well as there is a consensus sequence that was later discovered that is around the start codon, which is necessary to recognize the starting site for transcription. So it obviously contains the start codon, but it also has particular consensus sequence that are present on both sides. So depending on how big the letter is, that is the percentage probability that this is the particular nucleic acid uh, present in that location. So this was done after a lot of comparative studies between different start sites, and they found that there is actually a consensus sequence that is coming up that is similar across different organisms. The ATG is completely present on all these organisms, but the others can be a bit here and there, but they are a consensus sequence. This is known as the Kozak consensus sequence, and is present throughout in different organisms and types of cells. And the other thing that we are looking at is localization of mRNAs. So we are taking the example of nanos. So nanos RNA is present in mRNA is present in the Drosophila oocyte and is involved in symmetry breaking. So it determines which side will be anterior and which side will be posterior. And how does it work? So our nanos mRNA is transcribed and given out by the nucleus. It travels everywhere, but only in the posterior is where there is a different protein which starts binding to the nanos, and this protein makes sure that the nanos mRNA is only localized in the posterior. So bound to it, and then that other protein is also bound to the posterior region. So nanos is bound to the posterior region. Only the posteriorly present nanos will be translated. Every other RNA molecule of nanos will never be translated, and that is why we have a gradient being set up from the posterior to the anterior of nanos, and nanos in turn determines which side will be posterior. So this is how the entire symmetry breaking and posteriorization of the embryo works. There is localization of the nanos mRNA to the posterior region, which determines that this will be the posterior, and nanos after that takes over and then starts transcribing different genes in the it starts transcribing different genes which determine the posterior part of the oocyte. Okay, not posterior part, not posterior part of the oocyte, rather posterior part of the embryo. Okay, identify which of this following is correct. So the options are mutations in the Kozak sequence will hinder efficient translation. This has been shown. It is not only the ATG is absolutely required for translation initiation, but the Kozak sequence helps in efficient translation. And Everything else doesn't matter. The last option is also correct. mRNA localized to specific region within an embryo can be selectively translated, as we saw. Whenever only when nanose is bound to the anchor protein, which is bound to the posterior region, will it ever be translated, and that is why it helps in posteriorization of the embryo. Okay, moving on. A mutation on the X chromosome causes a recessively inherited disease. All children of couple A are affected by the disease. Couple B does not suffer from the disease. While one of their two sons suffers from the disease, the other son does not, and none of the daughters are affected. If the son of a couple A marries a daughter of couple B, what is the highest possible chance that their only daughter suffers from the disease? So this is essentially X-linked X-linked uh, essentially X-linked Punnett square. So I imagine that you have gone through the lectures and you know how to do it. So I will give you a minute to try and do this, and then we will solve it together if you are not able to. Okay, so that I hope is first enough time to figure it out. So let us look at it. We will move this even further. So given the information from the question, let us start looking at it. So if all the children of couple A have the disease, which is what the question means, then their son will need to have a genotype X-Y, because obviously if it is an X-linked recessive disorder, then it is on the X chromosome. So if their son is the one that we are interested in, so his genotype will be X-Y. If neither of the couple B have the disease and their daughters are also not affected, so as it says that the maximum probability that someone will be affected can only be derived from the situation when the daughter is also XX dash. So she is a carrier of the disease, but herself is not affected because she has the dominant allele, which is the wild type, and she is fine. The maximum chance of the disease is what we can draw from the Punnett square. So coming from the son X dash Y, from the daughter XX dash, do the Punnett square, you see that there is this there will be one daughter who is a carrier, one son will be fine in the F2 generation. This is one daughter which will have the disease and one son which will have the disease. The daughter has it because there are two recessive copies present. The son has it because the only copy he has is the disease copy. Since the Punnett square shows that out of two specific genotypes, so if their only daughter, the possibility of their only daughter inheriting one of these two is obviously 50-50. So since the Punnett square shows that at most one out of their two daughters will have the disease, the answer is 50-50. So you can either have this and you'll be fine, and have this or you will not be fine. So probability of the daughter having the disease is 50-50. Okay. Option A, answer is 50. Fine. Next, we have another genetics question. In a dihybrid cross between pure breeding pea plants bearing round and yellow seeds and wrinkled and green seeds, 100 plant bearing round seeds were obtained as F2 progeny. What percent of these 100 plants will have green seeds? And put in the answer. So this is a dihybrid cross. So I will give you like 2 minutes to try and solve this, because this is actually going to take a bit. Actually, I will, yeah, give you a minute. Okay, so now we look at it. This is the solution. I will completely remove this. First, let us look at the Punnett square. This is what the question is. So you have yellow, red, and green wrinkled, yellow round and green wrinkled. Both are pure breeding varieties. So you have the genotype is Y, capital Y, capital Y, capital R, capital R, small y, small y, small r, small r. This is basics. And the gametes in turn would be uh, capital Y and capital R from this plant, and small y and small r. And then we do a dihybrid cross in which you make the huge Punnett square out of it. So the square is not important because we have the actual information here. So having the yellow and round dominant phenotype, the probability is now 9 out of 16. Having the heterozygous phenotypes is 3 out of 16 each, because there are two heterozygous phenotypes, green and wrinkled, and round and yellow, or green and round and wrinkled and yellow. And these are 3 out of 16 each, and then 1 out of 16 is going to be the recessive, complete recessive phenotype, which is this one, which is 1 out of 16. So this is what we know, and this is what is written here as well. So you do this is the parental generation. The F1 generation is going to be YYRR, and the F2 generation is going to have the following. So parental generation you cross, you make a heterozygous in the F1 generation, and in F2 you self cross to get this huge Punnett square right here. And as we see, the dominant phenotype is 9, the heterozygous one has 3 and 3 each, and the green wrinkle, so this is the ratio, is only 1 out of the entire 16. So by the problem that we have, we know that the round seeds are going to be 100. That is what we have been told. So to have a round seed, you will have to have the proportion is going to be only these two kinds. So yellow round and green round are the only ones who are going to be the round seeds, and these the ratio is 9 plus 3 12 out of the 16, which is going to be the total. So 100 round seeds equal to 12 by 16, the ratio of round seeds to total seeds, multiplied by the total number of seeds. So now the total number of seeds is equal to just basic maths, 100 times 16 by 12, but I did not calculate because this is not what we need. We need green round seeds. So green round seeds is 3 is to 16. That is the ratio of the total seeds. So 3 by 16 into total seeds, this is what we plug in here, and just calculating gives us 25. So the number of green round seeds that we have is going to be 25. This is the entire calculation. You have to do all of this to finally answer the question. Okay, I hope this is fine, and this is all I had for you today. This finishes the quiz for the week 3, and if there are any questions, then you can just go ahead and put them into the comment on the YouTube section video when you look at it, and I will upload this presentation to the Google Drive. You can have access to it as well. Let me know if you have any questions. Okay, then I will say thank you for joining, and see you next week. Thank you.